The next type of biological assay is endpoint determination assay. These assays are performed when the characteristics of the indicator organisms are not suitable to perform or to measure the optical density or its turbidity. That means we can measure the optical density only when the particles are suspended in that particular solution in a homogeneous manner. However, there are certain microorganisms which are not able to form a homogeneous turbidity. Then the turbidity measurements by using colorimeter or spectrophotometer, it becomes difficult because it is not homogeneous. So in case if the indicator organism is having a tendency to grow in clumps or to grow at the bottom or to grow at the top forming a layer, then still the assays are possible by using endpoint determination assay. So whenever the turbidity measurement is difficult, then this is the option. Rest of the protocol is same. Here also you have a standard reference compound. So that standard reference compound is then diluted as shown here. For example, in case of antibiotic, then this each dilution contains now standard antibiotic, the growth medium and the indicator organism, that is antibiotic sensitive organism. Then this set of tubes, it is incubated for desirable time at desirable temperature, and next we'll get the response in terms of the growth of indicator organism. So here you can see it is the turbidity is not homogeneous, it is increase or decrease. So these tubes are not then exposed to colorimeter or spectrophotometer because we are not going to measure optical density here. There is simply going to be a visual observation where we can see that in last two tubes having 60 and 70 micrograms per ml of antibiotic are showing absence of growth. So there is no growth in last two tubes. This means that 60 micrograms per ml, this is the lowest concentration of that antibiotic that inhibits the growth of indicator organism. We can call it as minimum inhibitory concentration. So minimum inhibitory concentration is the lowest concentration that inhibits the complete growth of indicator organism. So in this case, it is 60 micrograms per ml. This is the standard set of tubes prepared using standard antibiotic. Now you have the sample. This sample, that is a fermentation broth in which that antibiotic is present. So that sample has to be diluted and added to growth medium plus the indicator organism. Here it is shown 3 dilutions are prepared of the unknown fermentation broth, 1:1, 1:10, 10 times diluted, and 1:100, 100 times diluted. Then these 3 tubes are then processed just like the standard set. These tubes are then incubated, and say for example this kind of pattern of growth is observed. You can see here that in first two tubes 1:1 and 1:10 there is absence of growth, and in the last tube, that is 1:100, that is having a least concentration of antibiotic, so it is showing the presence of turbidity. Now you have to compare this set with this set. Here we have determined that 60 micrograms per ml is the lowest concentration of antibiotic that inhibits the growth. We have to compare this with this. What do you see? Here also we need to see which is the highest dilution containing lowest concentration of antibiotic inhibiting the growth. It is 1s to 10. This means that this 1s to 10 tube contains 60 micrograms per ml of antibiotic. So this is the highest dilution and lowest concentration of antibiotic that inhibits the growth, 1s to 10. So that is compared with this set. So the final concentration of antibiotic in fermentation broth will be then. Now this fermentation broth is diluted 1s to 10. So what will be the dilution factor? It will be 10. So it will be dilution factor into 60. So 10 into 60, it will be 600 micrograms per ml. That means the original fermentation broth contains 600 micrograms per ml of antibiotic. This is how the endpoint determination assays are performed. Then there is another possibility that the compound, for example antibiotic, is a newly discovered compound. That means we don't have any standard reference compound. Still, turbidimetric assays, still the assays can be performed, which are called as end point determination assays. So whatever the newly discovered compound is there, that compound is first diluted. We can see the tubes here. First tube is 1s to 2, which is double diluted. Again there is double dilution 1s to 4, so on and so forth. The last tube is showing 1s to 128. This means that the first tube has highest concentration of antibiotic and the last tube has lowest concentration of antibiotic. That means the first tube is least diluted and the last tube is higher highest diluted higher diluted. Then just like turbidimetric assay, the samples are then mixed with growth medium that will support the growth of indicator organism and of course the suspension of indicator antibiotic sensitive organism. And then this set of tube is incubated at desirable temperature for desirable period, and after incubation see you may see this type of pattern. That the pattern is left hand side tubes will show very less growth or absence of growth, and the right hand side tubes will show the presence of growth, because left hand side tubes have highest concentration of antibiotic because it is least diluted, and the right side tubes are highest diluted and it will have the least concentration of antibiotics. So obviously wherever antibiotic concentration is less, there will be presence of growth. So in this pattern we can see the last tube, that is 1s to 128, is showing presence of growth, rest of the tubes showing absence of growth. So here the highest dilution of antibiotic which is inhibiting the growth is 1s to 64. So we can say that this tube contains antibiotic in a concentration of 64 dilution units. These are called as arbitrary units. Arbitrary means we have decided these units. Why we have decided? Because standard reference compound is not available. So these are arbitrary units. So in this case it will be 64 dilution units, which are called as arbitrary units, that is AU. So the concentration of unknown antibiotic in fermentation broth will be 64 dilution units. Then the next type of biological assay is enzymatic assay. It is different from enzyme assay. Enzyme assay means these are the assays in which we measure the concentration of enzyme, which is different from enzymatic assays. Enzymatic assays involves use of enzymes to measure the quantity of fermentation product. That is the difference between enzymatic assays and enzyme assay. You will be learning enzyme assays in biochemistry practicals. So it is different where we measure the concentration of enzyme. Here we are measuring the concentration of fermentation products with the help of enzymes. So in these assays, enzymes are used for the measurement of concentration of fermentation product. The specific features of enzymatic assays are, we know about the specificity of the enzymes. Enzymes are specific for their substrates. So enzymatic assays are highly specific and they quantitatively detect the minute amount of fermentation product. That means these assays are highly sensitive. Sensitivity means it can determine minute quantity of fermentation product too. So these assays are specific and sensitive. These assays differentiate between biologically active and inactive forms of a compound. That is also about the specificity of enzymatic assays. The protocol is an enzyme preparation. So enzymes can be obtained in pure form from a commercial source. It can be obtained from a microbial culture or microbial cultures which are lysed. That means the microbial culture lysate that will release the intracellular enzymes, or other enzyme source. For example, plant preparations or animal preparations or insect preparations. So this enzyme, under standard conditions, it is incubated with a sample of fermentation broth. So this fermentation broth may contain a mixture of compounds, but these enzymes are going to recognize or bind to only specific fermentation product of our interest. So for that, the enzymes are incubated with the fermentation broth. The enzyme substrate complex will form, and this will cause enzyme mediated change in the fermentation product. So there will be formation of ES, there will be formation of EP, and there will be formation of E plus P. That means this enzyme will cause some transformation into fermentation product, and it will convert it into some other product having different properties. So it can be that change that an enzyme can cause are partial decomposition with consequent formation of a product. So due to this transformation, the product will be formed. Then the amount of product formed is measured by some spectrophotometric assay or some any other assay. Then the amount of this product form is directly proportional to the concentration of fermentation product present in the fermentation broth. That is the principle and protocol of enzymatic assay. Now let us understand this with the help of an example. Assay of L-glutamic acid in a small sample of fermentation broth. So we are interested in measuring concentration of L-glutamic acid. So this fermentation broth so for this measurement there is requirement of an enzyme. That enzyme is glutamic acid decarboxylase. As the name indicates, the substrate for this glutamic acid decarboxylase is L-glutamic acid, and as the name indicates, that is carboxylase, it is going to release CO2 molecule. So the source of this enzyme is Escherichia coli. So these Escherichia coli cells are washed, which contain this intracellular enzyme. Then toluene is added to this mixture. Why? Because decarboxylase enzyme is an intracellular enzyme. Your substrate glutamic acid is present outside. So to liberate this enzyme from the cells, toluene is added, a chemical agent that will destroy the cell membrane and cell wall, and it will cause the release of intracellular contents outside. So glutamic acid decarboxylase will be released out in the medium, and then the preparation is incubated at desirable temperature at pH 5. So these assays should be carried out under standard conditions because enzymes are highly sensitive to physical and chemical conditions. So pH and temperatures are very important. After incubation, the decarboxylase will act on glutamic acid to release one molecule of CO2. So 1 mole of CO2 is liberated from each mole of glutamic acid. The CO2, which is poorly soluble in water, that is in the medium, at this pH value, it is evolved to the atmosphere as gas, and that gas is trapped in a chamber, and it is measured by manometric means such as with Warburg's respirometer, which is an equipment that will directly measure the quantity of CO2 generated. So as we can see in the third line, that is 1 mole of CO2 is liberated from 1 mole of glutamic acid. So the number of moles of CO2 will be directly proportional to the number of moles of glutamic acid. So this direct proportionality relationship will be useful for the measurement of concentration of glutamic acid in fermentation broth. So this is how enzymes are used for measurement of fermentation product. That is, it is a kind of indirect measurement of fermentation products. You can solve these questions on ion exchange chromatography and physicochemical assays and diffusion and turbidimetric growth assays.